too. So thank you, Holden. It's a pleasure to be here and present to you this little talk about the NanoPro immunoassays. And just like Holden said, some of you may find correlations to traditional technologies that already exist out there. I always tell people that NanoPro immunoassays are sort of a cross between a Western blot and a real-time PCR, although it's sort of like real-time PCR for proteins, not genes. So that's the difference. And this is just a little overview. I'm going to give you a sort of technical overview of the procedure, and then Dr. Fan is going to talk to you more about the specific applications for translational research or for clinical research. So with what I said, that NanoPro technology is sort of a cross between real-time PCR and Western blot. I'm sure most of you have done Western blots. You can find correlations to Western blots, but there are three main differences. And one of them is that this method is fully automated. So yes, you are trying to characterize proteins. However, you don't have to deal with any wet transfers to membranes. You don't have all those manipulations on the bench. You basically put everything on a plate, like in a qPCR technology, and then the platform called NanoPro 1000 will do all the manipulations and analyze it for you. The second main difference is what's called nano. We can analyze very, very, very tiny amounts of samples, nanoliter amounts, and so that translates to picogram amounts of proteins. So that allows you essentially to measure alterations to different drugs in very, very small amounts of clinical tissues. And I'm going to talk more in detail about it so you'll appreciate why this is so important. And the third main difference is that just like in the Western blot, you're trying to separate proteins based on size as they go through your electrophoresis. Here you're really separating proteins based on charge, and that allows you for more manipulation, so for more data richness, because not only do you find the expression of a given protein that you're interested in, but you can also detect specific changes in protein isoforms, and that gives you just much, much more rich data to identify biomarkers that you're interested in, or to look at real-time changes in giving patients therapeutics, and Dr. Fan will talk more in detail about that. So essentially the entire reaction is an automated process carried out in a tiny little capillary, as you can see here, which is loaded onto the machine called NanoPro 1000. So the capillary is filled with 400 nanoliters of your mixture of sample, which is your protein, and fluorescently labeled standards, PI standards, and amphiolites. And then you apply voltage, and when you apply voltage, some things go to the positive end, some things go to the negative end. So you can basically separate different isoforms, which are either phosphorylated or unphosphorylated, because the phosphorylated ones will go towards the acidic end, and the non-phosphorylated ones will go to the more basic end. So you'll have a nice separation after all this happens. And then there is a proprietary immobilization step where you basically shine UV onto the capillary, and that immobilizes the proteins that you have resolved by charge to specific places within the capillary. So you lock them into one place, and that basically allows you to then wash out the contents but leave the proteins there, and then do all kinds of different manipulations, such as incubate with specific antibodies that you're interested in, followed by your secondary antibodies, followed by luminol and peroxide, which will then cause chemiluminescent light to be detected by a CCD camera, and that can be captured. And the outcome is this. I mean, this is just a schematic, and I'll show you some real data as well. But you get this trace, which some people call electropherogram, and basically these peaks and valleys are, these peaks are where the specific isoforms of proteins are located, and then you can analyze, there's a specific software called Compass, you can analyze the areas under the peak, you can quantitate exactly, specifically normalize it to your loading controls, and then get very, very specific data regarding your experiment. So how is this carried out in practical sense? So you need a plate. It's actually a 384 format plate, but you only need a portion of it, which essentially corresponds to a 96 well. And once you have your plate and you have loaded the plate with your samples, with rows of antibodies that you're interested in and your detection reagents, you put everything in the machine. Like I said before, the machine will help you to separate the proteins based on charge and mobilize them to the right places. Let me just go back one step. So the NanoPro machine will draw your sample in, and then the immobilization step, the separation immobilization step will happen. And then you will essentially program the computer, which runs the machine, to draw antibody from the wells that you have indicated. And the proteins will be incubated with that antibody. And then just like in the Western blot, you'll follow this with a secondary antibody step. You'll incubate that, wash this out, and then you will follow that by aluminal peroxide combination of reagents that will generate your chemiluminescent signal, your shining light, and then you will be able to detect signal that will be captured by camera, and you can then proceed with your analysis with the Compass program. So there are several things that you can do differently when you set up this kind of assay. You can either load one kind of sample and then you can have several different kinds of antibodies, or you can do the reverse. You can have several different kinds of samples with one antibody. It's variable. It really depends on the user and your specific application. Essentially, in one experiment, you can use up to 96 capillaries. But in practical sense, people usually load maybe 2 or 3 or maybe up to 4 rows of samples, and then have the different combinations of antibodies that the samples can be cross-reacted with. So, yes?
So all these reactions, they happen within the capillary?
They all happen within the capillary. So you essentially start with the plate. You load the plate with the specific reagents that will be used for the assay, including your protein samples. And once you put it in the machine, all you need to do is program the algorithm into the computer, and that will drive the NanoPro platform to move the robotic arm in a sequence that you have indicated in your algorithm. So it's a very user-controlled method where you basically, I always tell people there are basically 4 steps to doing an NIA assay. The first step is truly conceptual. You have to sit down at your desk and plan out the experiment, plan out your plate, and plan out the algorithm, what kind of cycles, I call them cycles basically, the reactions that you're going to do and in what sequence. And that will allow you to program the computer in the right way. And the second step is the manual, basically doing all the dilutions, filling your plate with the right reagents. The third step is the actual running of the machine, which is out of your hands. It's basically 18 hours of the machine doing all different incubations. And then the very last step is you come back and you get your data and you do your analysis. And that's basically the 4 steps. So I just wanted to give you this. This is a very important slide because this will give you an appreciation for how powerful this method is. Because if you look at the more traditional ways to characterize proteins, the main challenge is that you always need a fairly large amount of sample input. So for mass spec, you need at least 100,000 cells to start with, and then with the further advancement of technical methods, you can go down to maybe 1,000 cells when you do protein arrays. But when you do nanopro assay, you can really go down to 25 cells, and that's a really small number. So for someone who is doing clinical work, and the samples are extremely rare and precious because they're either fine needle aspirates or biopsies from tumor, you just don't get more than what you have. This becomes very, very important and a really beneficial technology to analyze very small numbers of proteins. So another advantage of using this technology is the data richness, which I have mentioned before. So compared to the traditional Western blot, where you see the 2 bands up on top of this slide. So if you're, let's say, looking at your MAP kinase pathway and you're probing your Western blot with ERK, you're going to get this doublet that most people are familiar with. However, if you do an nanopro assay, not only do you get the expression of those 2 main peaks, ERK1 and ERK2, but you get everything else in between. So any phosphorylation that happens due to whatever treatment of cells you have done, or maybe a therapeutic that a patient got and you're analyzing those cells, you'll be able to detect those phosphorylated isoforms, as you can see in those peaks, and you'll be able to get very reproducible and quantitative data. I'm sorry.
Sometimes in Western blot antibodies are a little dirt, no? And how you can distinguish the real band from the, I don't know, signal that have not specific?
Right, so that's a really good question, and actually Dr. Fan will address that. So I don't want to elaborate on this too much, but this is a very important question that we deal with every day. So I'll let Dr. Fan answer it when we go on. So let me just move on, and just like I mentioned before, that very last step of your assay is essentially analysis, and there is a software that accompanies this procedure. It's called Compass, and it's very powerful because you can do all kinds of statistical analysis. And for those of you who want to see a little demo today in the afternoon, I'll be happy to show you exactly what it can do. But essentially, the main thing you want to do here is calculate the area under the peak, normalize it against the loading control, and then you will get very specific, very numeric, quantifiable data, which cannot be done with a Western blot, even if you get your bands and then try to quantitate it with VersaDoc or whatever other method that you're using. You just can't quantify it because of all the transfers you have done, and there are just too many variations. So I just try to list all the main advantages of the NanoPro system, and I'll go through it fast because I've already explained those. So one, it's just to put this in perspective as a summary. One is the ability to assay precious or quantity-limited samples. You can go down to very, very few numbers, like 25 cells, so picogram levels, really. It's data richness, so you can detect multiple phosphorylation states with just one antibody. So, you're using one antibody, but you're answering several questions. And it's high throughput, it's highly automated. So you can run up to 96 samples per run. Because it's automated, it's reproducible, it's consistent. There's less human error associated with the technology. And you can easily quantitate your measurement because of the Compass software. Now, I don't know why this is not. There we go. So this is just a little example. Dr. Fenn will elaborate on this, but let's say that you are studying the MAP kinase pathway, and this would be your typical trace that you get from the assay if you're looking at ERK. And so these are your major two peaks, ERK1 and ERK2. But let's say now that you are trying to treat the cells with a certain activator, or that you are looking at patient samples from cancer patients, and you can, with one antibody, see phosphorylation peaks. And so you can now see all kinds of different isoforms that come up. And so this is a really powerful method because you can look in real time what happens during activation of a certain signaling pathway. So this is just to give you an appreciation of all kinds of different antibody assays that have already been validated, and this is by far not a complete list because we always do new assay development, and we have several more that have been already validated by us. For those of you who are interested in getting a general sense of how much it costs, the way that we look at the cost is we charge the user by capillary, and the cost of capillary is the capillary itself, but also the reagents that are needed to run the capillary. And then there is essentially a time that is needed to run the assays, to set it up, to analyze it. So for a typical kind of experiment, and this is just an average, and it's going to be variable based on what the user needs and how many samples, how many antibodies you're going to run. But let's say just a very typical average assay where you're using 60 capillaries, that's going to cost you around $1,000. And there's going to be a slight increase if you need a tertiary step to amplify your signal, which I didn't really talk about, but there are ways to amplify, just like in a Western blot and other methods. If you do biotin, streptavidin step, you can amplify the signal several fold. And just to acknowledge all the people involved, definitely all the HIMC people, Holden, Alice Fan is here, who is both part of the Filcher Lab as well as the core facility that I'm running. And I'd also like to acknowledge Deba Brita de Basu from Protein Simple company. Protein Simple is the company that actually that manufactures the NanoPro 1000 platform, and they've been very helpful in collaborating with us in this effort.